of the cells in the presence of different kinds of concentrations of KCl, and also I was in the current clamp configuration, I was able to measure the action potentials that was very spontaneous. I didn't give any kind of stimulus, and these cells already were spontaneously triggering the action potentials. What was very interesting, because since they are mesenchymal stem cells, they should not have action potentials. But in this case, here are the cells. I think they have some neuritic shape, so maybe they are already differentiated in neurons, I don't know, but I was able to measure these action potentials. And beside that, I was able to do other kinds of configurations just to show you all what patch clamp can do it. You can measure the single channels like I showed you before, and here is the voltage clamp, where you have a pulse from -60 to 80 millivolts. We have the control cells when the basal conditions of the cell, and here in the presence of KCl, in this case we have the efflux of potassium from the cells. That would be the hyperpolarization, or even better, the repolarization of the cells after an action potential. So this is the technique that we use the most here in the lab. We are giving this voltage clamp for our cells, and you're measuring what's happening with the efflux of potassium of these cells. That usually comes after the influx of sodium or calcium. After the depolarization of the cell, we usually have the repolarization of the cell, which it is the efflux of potassium of these cells. So I'd like to show you this figure because I started my master's degree here in the Department of Physiology in 2002 in collaboration with Dr. Laura Leite, and this is the equipment we used to have in that time. So this is very interesting because we were measuring intracellular recording at that time, and with the intracellular recording all you can do is to measure depolarization and hyperpolarization initiating from a resting membrane potential of the cells. So it was a very simple experiment. You cannot do much with intracellular recording, except by the fact that it's very physiological. You cannot know for sure which channel are you working. You can use pharmacological drugs, but still doing patch clamp, it's much better. So after doing my master's degree, where I showed the effect of thyroxine in the plasma membrane, I decided to do my PhD, sandwich PhD outside of Brazil, where I should then learn the patch clamp technique. So in our lab, we always used to work with rapid responses and non-genomic responses, so independent of protein synthesis and gene transcription. So since I was always interested in learning what is happening in the plasma membrane, patch clamp is the best technique to do that. So that's why when I decided to go to United States, getting the University of California in Riverside, I started to study the rapid responses from vitamin D that happens very fast, in seconds or minutes, different of genomic responses that could be in hours or days. So these effects, they can trigger physiological responses already, or they can go through gene transcription after that. And this is when I studied for 2 years in University of Riverside. In this university, I showed the rapid responses because the vitamin D receptor, they have two different targets, the genomic pocket for genomic responses and the alternative pocket for non-genomic responses. So I showed in my PhD there that the 125 vitamin D3 binds to an alternative VDR pocket in the plasma membrane of Sertoli cells, and this activates the PKC and the PKA kinases that phosphorylates the chloride channels and activates the secretory vesicles to release different kinds of proteins that are important for secretion for the maintenance of spermatogenesis. So here is when 125D activates rapidly the influx of chloride inside of the cells, and in the end of the experiment I showed that the IDS, that's a chloride channel blocker, was able to block all the experiment. So this is just to show you one of the work that I have done with patch clamp for the first time in my PhD. So after that, I stayed for a long time in the United States doing calcium imaging that I'm going to show in the end of this presentation. But after that, I came back to Brazil And with the help with Dr. Vander, Professor Vander, we built up all this new lab here at UFSC. It calls Nubiocel, and this lab will have set up for studying the isolated cells from cells culture. That is what we do the most here in the lab. But we also have the setup for studying brain slices and doing calcium imaging. That is the setup here that we are just starting to work with it right now. So this is the two possibilities you have here in the lab. It's an institutional project, so everybody have the access to work with this if the person is able to do it, the technique, because it's very sensitive. So you should be specialized in that. It's not only getting there and doing some recordings, not so simple like that, but everybody can have the access to study the electrophysiology studies of any kind of cell possible. So
can I have a question?
Sure.
Please, if I have detached
if the cells are not attached to the plate, it's impossible to do the experiment.
Okay.
Was that the question?
Okay.
Yes, I have many experience with that. I already tried to work with them, but they run away from you. You are not able to reach the cell because it's going to leave.
Okay.
You know, so it should be attached. If the cells don't attach it, you can find a way to attach them with polylysine or something like that.
Okay.
But not always they're going to attach.
Okay.
So if you have a perfusion system, it's even more difficult because the perfusion system is going to run away with your cells if they are not attached.
Okay.
But if you have a system that you don't perfuse the solutions, if you are lucky, you can maybe try to get a cell that's not very well attached. But this should be attached, it's going to make your life easier.
Okay.
So here is just to show you how important it is to study the voltage-gated potassium channels, because that's the channel that I'm working the most now. I work with chloride channels, but now I'm working with potassium channels. And as you can see here, the potassium channels, they are very important for neuronal and cardiac action potentials, to regulate calcium signaling, cell volume, driving cellular proliferation and migration. They offer then tremendous opportunities for the development of new drugs to treat cancer, autoimmune diseases, metabolic, neurological, and cardiovascular diseases. So just to show how important it is for new therapeutical targets to study the potassium channels. So now I'm going to show you some data from the lab. So since I came back from Brazil, from United States to Brazil, we start to culture the Sertoli cells. As I told you before, the Sertoli cells here in pink, they are very important for nutrition support and the release of important proteins and all the fluid for the sperm, for the germ cells that's going to produce the spermatozoids. So Sertoli cells is very important for spermatogenesis and for male fertility. And we already showed that vitamin D binds to an alternative pocket in the plasma membrane, and I show you that activates chloride channels. But here I had the chance to show that vitamin D also activates the potassium channels, which is the efflux of potassium. And then we add in the end of the experiment tetrethylammonium, that's a broad spectrum blocker for potassium channels, just to show that we're really working in potassium channels. So this is what I've been doing with Sertoli cells. I tried many kinds of agonists. After 1.5D, I also used the reverse T3. Reverse T3, we got a very interesting data with reverse T3, because reverse T3, it's known in the literature to be an inert hormone. That means it doesn't really have any physiological action. But now you can find something from the T3 reverse. T3 reverse can actually bind somewhere in the plasma membrane, because not only me but other people found that RT3 binds very quick somewhere in the plasma membrane and trigger the efflux of potassium ions from these cells. And it was very interesting because the student that is actually doing this project, she showed that reverse T3 activates the calcium influx, the 45 calcium influx, which is a radio label calcium that you can measure the quantity inside of the cell. So first she showed that reverse T3 activates the influx of calcium, and then I showed that RT3 activates the efflux of potassium. That's probably after the depolarization caused by calcium, we have the repolarization, which is the efflux of potassium from the cells. And this is very interesting data. So after that, I had a chance to work with a different kind of cells. They call HEC293 cells. They are very used in the whole community of researchers, investigators. They are used for expressing different kind of proteins in the plasma membrane. So this is very nice because this lab that I'm doing a collaboration here at UFSC, they express at BK channels in these cells. BK channels are potassium channels that are dependent on intracellular calcium. So this is very nice because since I was doing this collaboration, here are the HEC293 cells, I decided to try my hormone there. So I added reverse T3 in this system, and very quick, like 1 minute, RT3 activated the BK channels of the cells expressing HEC293. I mean, the HEC293 expressing BK channels. And then I wash out the hormone and I block it with the potassium blocker in the end of the experiment. So what I showed here in expressed BK channel cells, it was very interesting because corroborated with the data I got in Sertoli cells. Besides this collaboration, I had another collaboration with another professor that works with Sertoli cells, that she works with ATP. ATP usually binds to the P2X7 receptors. These receptors are ionotropic, so once the ATP binds to the P2X7 receptors, there is an influx of positive charges inside of the cell that could be sodium, calcium or potassium, especially calcium. And then it was very interesting because I found that ATP not only activates the efflux of potassium, which is a washout and then block it, but also ATP is so potent that I was able to measure the depolarization effect of ATP. So here I have the resting membrane potential of Sertoli cells, minus 50 millivolts. Whenever I added the ATP 1 millimolar, I found a huge depolarization in the presence of ATP, and when I washed it, the cells, the effect was gone. Why this is important? Because this is the first step of the RT3 effect. First, RT3 activates a depolarization that we don't really know if it's calcium or sodium. We believe it's calcium, but after that the cell is super depolarized, and what happens? The repolarization of the cells, which I measured here with the efflux of potassium from these cells. So this should come first, depolarization, and then here repolarization of these cells activated by ATP. So this is very nice because this professor, this student actually, it's a PhD student, she's showing that ATP is very toxic for the cells and activates a lot of kinds of oxidative stress and things like that. So this is another project we're running in the lab. And also I added two other compounds in the cells, which is very interesting, but I didn't bring it today because it's exactly the same data. It causes a huge depolarization of the cells and the flux of potassium, which is ethanol. Ethanol is very toxic, as we know. So this project is very interesting because the student is giving ethanol for the animals in the lab, and we can see that it's very bad for the development of the mice in the lab. So this is just showing in vitro what it was shown in vivo, that not only ATP but ethanol and also Roundup is very toxic for the cells too, and Roundup, as much as ethanol, causes a depolarization. I mean, a depolarization showed like this, and after that a repolarization of the cells. So this is what you have with Sertoli cells. And then another project that we're running in the lab is potassium channels in beta cells from pancreatic islets. So just to remind you first, when you have glucose entering inside of the cell, you have the increase of ATP concentration. ATP is going to close the voltage dependent, the ATP dependent potassium channels. Then we're going to have a depolarization, and it's going to open the voltage dependent calcium channels. the calcium is going to activate the insulin release, and then in